Okay, so the next question is, are there T cell or B cells or their receptors really recognizing this mutation? We mentioned that the T cells and B cells, they have these type of receptors that recognize, all sorts of non-selves. The question is, each of us only have one copy of mom's DNA and dad's DNA. So T cell receptor gene, we only have two, one copy from mom and one copy from dad, but we can evolve to have T cells and B cells with many different receptors to recognize different antigens. And this is through a process called the VDJ recombination. This is what happens in the T cells. You have this really long gene segment in here, and there are many V's and many D's. So the D is orange and many J's. These are blue. You see there another one. The V's are together. Each D has a number of J. This D has a number of J. And so in the VDJ recombination process, there are like DNA cuts in the genome, and things in the middle get deleted, and then things are stitched together. And so it always happens that there are basically one V, one D, and one J will be combined together, and the closest VDJ here is able to make a T cell receptor. So for example, if this V is being selected, the V before it will not even get a chance to make a T cell receptor. Whereas if, in this case, if this this J is being selected, the next J will no longer be functional. And so every T cell have a different combination of VDJ, and that combination creates the diversity. In addition, anytime you cut the DNA, you try to stitch them together, that junction sequence, there's no guarantee how they are going to repair. And so very often the junction sequence will have small insertions, deletions, and also mutations. That's this yellow region. That's the VD and DJ junctions. And there you create a lot of small insertion, deletions, or mutations. That's different from your original DNA. And by the way, this actually happened on the DNA level. And only the closest VDJ will make a productive RNA, then make a protein, which is the T cell receptor. That's the T cell receptor, which is presented onto the cell surface. And then that will present antigen, which will be recognized by the T cell receptor will recognize the antigen being presented by the antigen presenting cells. This is happening both on T cells and also on B cells. You can see here again, V, D, J. We randomly, well, not we, but the B cells will randomly pick a V and D and J and stitch them together. And the closest VDJ will be made into this antibody sequence. And you can see here, these are what we call very hypervariable regions, and they are responsible for recognizing the antigen. And interestingly, in the T cells, once the T cell recognizes the antigen, it will clone itself. They make multiple copies of itself. It will get activated, and it will also start the killing effect, which is secreting cytokines, and also try to punch the hole on the cancer cell and kill the cancer cells. For the B cells in here is after the B cells are activated, very often the B cell will make a local factory called a tertiary lymphoid structure. It's kind of a local B cell factory, and then it will make multiple B cells. And in this process, it will continue to get mutated on this hypervariable sequence, which is on the productive VDJ. Additional mutation will happen. So as B cells are being B cells are proliferating, they have more and more mutations. And the additional mutation, if it's able to bind to the antigen even better, that copy of the B cell will have even better signals to proliferate. And so it's kind of a natural selection in this little B cell factory. Only the B cells that recognize the antigen better will get a stronger signal to continue to proliferate, and then they will create a new mutation. If that new mutation is recognizing the antigen worse, they will not continue to proliferate anymore. But the ones that really recognize antigen better will get more proliferation. In addition, the constant region of the antibody will also shift. You can imagine, depending on the different constant region, some will get help from macrophage, some will call natural killer cell, some might call T cells. And so it's like when you call 911, you'll get policemen, you'll get firefighters and ambulance, right? So depending on the constant region, they also call different help. And so that's what happens in our immune cells once they encounter the antigen. And so it's a very, very hot area to know what are the T cells in the tumor that's recognizing the antigen, especially if we can know what they are trying to recognize. For example, this is all the V genes in the T cells in one individual. Sorry, here. Remember here's the V, D, J. So in the V, there are many, many different copies. And at the end, every T cell pick one D. But if you were to sequence a bulk tumor, you will see that this V is being picked a lot, and this V is being picked a lot in different cells. But other Vs, well, at least in this patient, somehow the other Vs don't appear so much. That this is from a different patient. And so you can see different Vs are definitely coming from different T cells. And so for the V, we can map it because the V fragment is fairly long. You can map the whole thing. But actually, the hypervariable sequence on this junction, the VD and the DJ, this region called the CDR3, this is the really very different hypervariable region between different T cells. They are responsible for recognizing the antigen. And because of this VDJ recombination creating the junctions that are not, small insertions, deletions, they can't really be mapped to the reference genome. And so interestingly, if you were to do a bulk tumor RNA-seq, this sequence is being considered as garbage. And not, the first thing you do, STAR or Callisto, like a salmon, you map the reads to the genome. This region is being thrown out because it can't find it in the genome. And so we actually, in the lab, developed an algorithm that really look for the sequences, especially, for example, for TCGA data. Most of the data are done in paradigm sequencing. And so sometimes the V sequence or maybe the J sequence can be mapped to the genome. But the other end, if it cannot be mapped to the genome, you know that it's coming from this region because of the VDJ recombination. And so we can actually, if there are enough same TCR that's sequenced with the same sequence, you will see that they overlap with each other. Then we can de novo assemble the VDJ sequence back from the original sequence. And so we have an algorithm for that, and we also run this across all of TCGA tumors. And on every TCGA tumor, we can identify maybe 100 or maybe a few hundred TCRs from the tumor. And then we can look at, what are the diversity of the TCR in the tumor. And this is something that is in one of our earlier papers. You can see the tumors on the left side, those tumors have overall lower mutation burden. And if you look at their overall T cell diversity, they also have lower diversity because you only need 1 to 2 type of T cells to recognize that mutation. Whereas on the right, whether it's because of the smoking or pollution, or whether there are carcinogens or radiation, or infection microbiome, these tumors have overall higher mutations. Their tumors, T cells, also have higher diversity. They have different TCRs potentially to recognize the very different mutations onto the tumor. And so interestingly, a lot of these right side of tumors, they respond a lot better to immunotherapy. You can imagine, because they have more mutations, they can be recognized, or if you just wake up the T cells, right, they are likely to at least some copies see something wrong with the cancer cell, then they can kill the cancer cells, right? So this is one study from Steve Rosenberg's group. They found a specific T cell receptor that is recognizing a KRAS mutation. So KRAS mutations currently does not have a very good small molecule inhibitor because the enzymatic pocket for KRAS is on a flexible region. And so it's very difficult to develop a drug that perturbs the structure. It's already a intrinsically disordered region. But the scientists in this study were able to find a T cell receptor that recognizes the mutant KRAS. So you can imagine if they make a special army of T cells specifically targeting this KRAS mutation and infuse those into the patient, then those T cells can just recognize the KRAS mutated tumors, which would be fantastic, right? So previously we discussed the CAR T. So the CAR T, this receptor is kind of engineered kind of antibody that can be recognizing some antigen, then get activated to make the T cell kill the cancer cells. But this is kind of an artificial system. The TCR-T, because all the T cells naturally already have a T cell receptor. Ideally, in this case, they have a natural T cell from the patient that recognize the mutation. You can also engineer the TCR onto the T cells. Then they will also recognize the mutations. And so in the T cell therapy world, there are some companies doing CAR Ts, other companies thinking about doing TCR-T as well. And recently, 10x Genomics have a new technology. You can do single-cell RNA-seq and a single-cell TCR-seq on the same tissue. And so this is a paper that's published this year. On the left side, you can see these are all the T cells from this patient. And based on the gene expression, they can divide the T cells into different subsets. You know, this one is a proliferating T cell, a memory T cell, or effector T cell, or exhausted T cell, or progenitor T cell. They can divide into very fine subgroups. And in each group, and then from the same T cells, from the cell barcode, they can also get the TCR sequence from the same T cells. So they can see what is the T cell in this, you know, what's a TCR clone in this population or another population, and so on. And so you can imagine if this is an expanded T cell clone, and it also secretes cytokine, right, the killing cytokine to kill the cancer cells, okay, those might be the specific T cell that is recognizing the tumor antigen, right? So you can learn what TCRs recognize tumor antigens and how do they evolve. What happens in the adjacent normal? Are these T cell clones coming from the blood, or are they coming from the adjacent normal, or are they already in the tumor that are just being waken up by the immunotherapy? And so using this TCR sequencing as kind of a clonal evolution, or at least to trace which T cells are coming from where. Are they also in the adjacent normal? Are they also in the blood? You could understand how immunotherapy really work. Of course, right now you only know the TCR sequence. To really know what they recognize in the cancer, that's still an open question. I think computational biologists are trying to do this modeling to see, for example, if this mutation, whenever this mutation happens, you see this TCR clone, oh, okay, then you see enough tumors, maybe you can match them together. But it's still, you know, so far the data is very, very limited. We only know a few of those T cell receptors that really recognize specific mutations or tumor antigens onto the tumor. Okay, questions? I think, you know, for the time, maybe we'll stop here and we will continue on the B cell recognition of the tumor antigens in the next lecture. Okay, thank you guys.
Thank you.